Alright, awesome. So today, as I said, we're getting straight to it. We're going to do some monohybrid drawings. Oh, that's a little too small. So we're going to do some monohybrid crosses and just to show you guys how it actually works, essentially. Hopefully we can get over around 5 questions, whatever. But yeah. So I'll show you how you're supposed to get all 5 marks on your, well, supposedly your test. But yeah, so it's kind of important. So we start off by reading the question. Obviously, in dogs, we have wire hair, and it's dominant to smooth. So they actually did it for you. They tell you that W is the dominant, but usually you would start off with a let statement. Why is not writing? What? Let W be wire. Okay, now what the hell? Wire. Okay, I can't write. Oh wait, I could have just done this. Okay, let me delete that. Okay, so let W be wire here. Awesome. And wait, you know what? Hold up. We'll delete everything. Oh, not that. Okay, so let's do. Let W be wire here and let W be smooth here. So we have the capital, that is the dominant one, and we have the recessive, that is the small one. So, in a cross of a homozygous wire-haired dog with a smooth-haired dog, what will be the phenotype and genotype of the first offspring generation? Okay, so this we're able to paint directly. So we're going to assume, and we're gonna look at the parent first. So I'm gonna zoom in a little bit here. So let's draw the parent first. This is P1, and this is P2. So remember, they are homozygous wire, so that means they have a big W and a big W. And for our P2, they have a small W. Okay, so this is where it comes into play. I really don't recommend you choose these letters that look very similar, both capital and lowercase, because it's just hard to distinguish. And it makes it harder for yourself, but whatever, the question did it. So the parent, we're gonna they have a big W's, small W's. We're gonna identify which one are they able to pass on, and we're gonna draw a circle around them. So we're gonna do the gamete. So identify. So this one for parent one, they can only pass on a big W because that's what they have. And for the parent two, obviously they can only pass on a small W. They don't look much difference, but that's not what I meant. That's not what I meant to do. Okay, so remember that you're going to have a cross, and you're going to have a cross. Obviously you're gonna get this from dad because this was the only thing you can get. This is the only thing you can get, and this is the only thing you can get from mom. So it's gonna come down here, and it's gonna give us W. So that, well, this is a genotype. So remember, the genotype is the genes. Genotype. No type. Nope, can't fit it in there. So this was a gene. However, we were also told that wire-haired dog was dominant to smooth-haired dog. So meaning that wire will completely mask the smooth hair. Meaning that this is going to be a wire-haired dog. Wire. And that's the genotype. Okay, now let's see. What would be the ratio of wired-haired dog to smooth-haired dog in the F2 generation? Okay, so often when the question doesn't tell you at all, it usually just means that, hey, you know what? It's time to do. It usually just means that you're self-crossing. So basically you're doing W W and W W. And can I delete all of this? Oh, I can't do it. How do I do this? Alright, so I'll just do that. So okay, a little bit forward. So now we have a new generation. This is going to be parent 1. Oh, I can't draw now. Parent 1, which remember was big W, small w. And parent 2, since we're self-crossing, we're doing a big W and small w. So now this is where it gets interesting, because the dad, or well, whatever, the mom, it could pass on a big W or it could pass on a small w. And same for the parent too. Big W, small w. Remember, you're only doing a single trait, so this is still a monohybrid cross. So let's cross. So now this is where you fill up a square, and we usually refer to this as a Punnett square or a Punnett square. So we put in P1 here, and we put in P2 here. And now we cross. So what does P1 have? P1 has a big W and a small w, and P2 has a big W and a small w. So now we're going to fill in this square. Looks like that. So this is big W, big W. That comes from this one. This one is a big W, this one's a big W, this one's a big, that one's a big W, this one's a small w. So we're going to do this. And this is a small w, this is a big W, W, W. And this is a small w, that's a small w. So W, W. Now let's identify what they are. So you have the ratio of the wired hair to smooth hair dog. So remember, these three, even though they are heterozygous and they have a smooth gene, because smooth gene is recessive to a wired gene, it means that they're going to be a wired hair dog. So that so that right there is a 3-to-1 ratio. And why am I not drawing on the page? Okay, whatever. So yeah, so that is a 3-to-1 ratio, because when you self-cross, you're gonna produce 3 wire hair dogs and only 1 smooth hair dog. Let's look at another question. Okay, let me paste it in here. Okay, right here. So we have wood rats. Let me delete this. Okay, I can't delete it, but I'll just delete it right there. Why can't I delete this? Delete. Okay, so wood rats are medium-sized rodents with lots of interesting behaviors. You may know them as pack rats. Let's assume that the trait of bringing home some shiny object. Wow. So bringing home some shiny object is H, capital H, and it's dominant to the trait of bringing home some weird dull objects. So now, suppose 2 heterozygous individuals are crossed. What will be the phenotype ratio and the genotype ratio of their offspring? So let's do P1, and on the other side, we'll do P2. So remember, they're both, what are they? Heterozygous. So it's a big H and a small h. And that's the same for this, big H and small h. And we have, so this was step 1. All right, so actually, no, this is not step 1. Your step 1 should be let the letter, whatever letter you choose, represent the trait. And then the step 2 is to draw out the parents. What do they have? And then step 3 is to draw out this. They could pass on the big gene or the small gene. And for the same thing here, big gene, small gene. I know that looks kind of bad, but I don't have good handwriting. So anyways. So let's do a Punnett square. So P1, remember, it goes here. And P2, remember, it goes here. Or, okay, you can do P2 up there, but it doesn't matter. So big H, small h, big H, small h. Now we cross. So H, big H, big H, big H, small h, big H, small h, H, H. So since it lasts for the genotype ratio, we're gonna write it out. Right, we're gonna write it out for them. Geno, and we're gonna write out pheno here. Pheno. So for genotype, we have this is full homozygous dominant. That's 1, and that is equivalent to 2 heterozygotes and 1 homozygote recessive. So 1 to 2 to 1. And phenotype, remember, all of these 3, they like bringing home shiny objects, and this one likes bringing home dull objects. So that is still a 3 to 1. And we're done. The entire question is over. So as you can see, okay, hold up. So This let statement, that's 1 point. Identify parents, gamete possibilities, that's 2 points. 3 points for doing a Punnett square. 4 points for getting the genotype, and 5 points for getting the phenotype. So remember, you're getting all 5 points. Well, you want to get all 5 points, but sometimes it's not possible because your teacher is bad, but whatever. So you're trying to get all 5 points. And remember, some of these questions could be 4 points if they ask for different stuff. Or they could be more points if they ask you a question. Like the one that I'm about to show right here. Let me undo everything. Okay, I can't. Okay, now I gotta Jada click it. Don't I? Right. Okay, why can't I just add another layer? I can't. So sad. Okay, so we have our new question here. Obviously. That one's gonna delete, that one we're gonna delete. So it's gonna be the last question because I don't want this lesson to be too long. It's a pretty simple lesson. I figured everybody would be able to get it pretty easily. So there's this cacti called saguaro, whatever how it's pronounced. They are very tall cylindral plants that are usually have 2 L-shaped arms, one on each side. Suppose you live in southern Arizona, which I don't. The cactus is common, and you happen to have one growing in your yard. Very lucky. Your cactus has 2 arms, but one is longer than the other. Now, let's assume that arm length in these cacti are controlled by a single gene, with the same arm length, A being dominant, 2 arms of different length. Okay, so same length. Okay, you know what? Let's do a let statement this one. Let's do a let statement on this one, so we're getting all 5 marks, because I don't want to lose mark. Let A be same arm length. And let's drag a little bit this. Let's move it a little here. Okay, let A be different arm length. Alright, so now we have the let statement. That's 1 point right there. We just earned ourselves. And let's look at the parents. So, parent 1. I'm going to do P1. What is the genotype of your cactus? So, well, hold up. We're going to identify this. We're trying to get our points here. We're not trying to do the cross and everything. We're trying to get points. We're trying to answer the question. So, what is the genotype of my cactus? So, my cactus has 2 arms. So, they're different lengths. So, remember, recessive. If you have one single dominant trait, then the recessive is going to get masked. And the phenotype, remember, is looks. Alright, phenotype always looks, and genotype is always DNA. So our cacti has a different arm length. So that means we have one big trait and 2 small traits. So that is the genotype of my cactus because it's recessive. Could one of the parents of your cactus had a phenotype with arms of the same length? So here we're bringing in the idea of what are we actually doing. So I didn't go into too much of this, but test crosses. So this is where we bring in the idea of test crosses. So let's say I have this children. I don't know what his genotype is. I can observe his phenotype. He could be whatever, but I don't know what his actual genotype is. So we want to look at his parents. And if we know the genotype of his parents, we could potentially be like, hey, they have this chance of producing this children with this amount of genotype. So that's where we bring in the idea of test crosses. So now we're going to perform a test cross on the parents. So let's say one of the parents had a phenotype with arms of the same length. So phenotype, remember, this is phenotype. It's the looks. It only looked like that. Maybe it had a different length gene. We don't know. So obviously let's drag it down here. For this, for our baby cacti to have 2 small genes, it means that both parents must both had a small gene. So right there, we can get it down. Both parents must had a small gene, P1, P2. So now they want us to tell them, is it possible that one of them had a phenotype with arms of the same length? So we gotta add a big here. Let's add a big A. So now this is the arms of the same length. And let's just say that P2 was also the same thing. So now when we look at the genotype of our cacti, there is still a good 25% chance of him getting this gene from mom and this gene from dad, because we got to do a cross now. So A, 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 A. And now that looks like A, 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 A. So we have still a good 25% chance of getting it, even if both of our parents was heterozygous. So that answers the question. Yes, one of my parents or both of my parents could have had a phenotype with an arms of the same length, yet still produced me, because they could have been the heterozygous. So here, the only thing that they want you to mention is probably the idea of heterozygotes and the idea that you have a 25% chance of getting that gene. And that was pretty much it. Now let's go on to the last question. Wow, this thing is being stupid. Suppose you cross your cactus with my neighbors, which has arms of the same length. Your great-grandchildren. So I probably died by then. Unfortunate. So my great-grandchildren, he takes the sacred cactus a long time. Okay. It finds that half of the resulting offspring have arms of the same length, and half of the arms have different lengths. So what was the genotype of my neighbor's cactus? All right. So this is test cross even further. So we know that our cactus, it's supposed to be fully recessive. It's small a and small a. Why did it pop out again? So small a and small a. So it crossed with something to create half the offspring. So half of them had different length. Okay, so half of them had different length. So let's say they produced 4 offspring. So half of them were this. And because this is both recessive, the only thing he could pass on is an A. So means the rest are all heterozygotes. Alright. So now we look at this. So first, what we can know is that the neighbor must, suppose, what was the genotype? Okay, so we have to figure out the genotype, not the phenotype. The phenotype would have been so easy. So the neighbor must have the big A, because for the children of our cactus to have a big A, it must have came from the neighbor, because our cactus cannot pass an A. It cannot pass something it doesn't even have. So obviously the neighbor had one. So now let's just say the neighbor was fully heterozygous. So let me do something. Let me do this. The neighbor was full. Wow, that's ridiculous. So neighbor was fully heterozygous, and the only thing you could pass on is obviously the big A. Sorry, homozygous. So let's do a test cross and see what are the chances it's going to be. So A. So it means that if the neighbor had a full homozygous, and what was that? It was arms of the same length. If it was homozygous arm of the same length, then all of our children will have a 100% of being a carrier, which is basically arms of the same length. However, that was not the case. So our children, it had half of them was different length, and half of them was the same length. So what does that mean? That means this. It had to be a small a. So now we had a big A and a small a. Now, remember, we're doing test crosses. We can change the genotype of this neighbor, because we don't know it, and we're just doing theoretical stuff. So it's not unethical when we do it on paper, but it's pretty unethical when we do it in real life. So let's do it here. A, which is what we have here, and we have A, which is what we have here. And look at this. 50% chance, 50% chance. And that answers the question. The genotype of my neighbor's cactus was a. And your conclusion statement. And yeah, I'm about to end it here, because monocrosses is easy, and I want you to, so the big takeaway of this class is there's this thing called monohybrid crosses that you need to know how to do, and they're extremely easy, and there are also something called test crosses, which we can use to determine the unknown genotypes of your close relatives. And when I say close relatives, I don't mean like your maybe not your grandparents or your cousin or your uncle. I meant your direct relatives, your dad or your sister or your mom, whatever. However, yeah, so that is probably it for this class.